In ORATOM, my main tool is ORATOM-MD simulations. In ORATOM-MD simulations, we define a biological system, energy of biological system using as a function of the position of every atom, including hydrogen. So that's why we call our technique ORATOM-MD simulations. When the physical energy function of the system is defined, we can just solve classical Newton's equation, F equal ma, using powerful computers like Bluewaters. Then as a result, for example, this one is a typical example of protein folding, which is one of the original and one of the most important problem, target of the MD simulation study. This is a quite small model protein called the WW domain with only about 35 residues. But using ORATOM simulation, folding this model system, model protein, used to be challenging until about 5 years ago. But now, if we have only about 1,000 load hours on Bluewaters, anyone can do protein folding simulation really easily. Not easy, but but the today's topic is not about protein, but about DNA. DNA consists of 3 functional groups. Phosphate group, which is shown in red, negatively charged, and sugar group, and the DNA base. DNA base, there are 4 different DNA bases, ATGC. So by combining ATGC sequence in a row as a polymer, one can have a single-stranded DNA. And by combining 2 single-stranded DNA molecules, we can have DNA helix, double helix. And DNA double helix is the fundamental unit of our genomics. DNA double helix has about 2 nanometer diameter. But in our real human genome inside our nucleus, we have billions of DNA monomers in 46 different chromosome fibers. And as you can see in this model of chromosome fiber inside our nucleus, that has only about 10 micron diameter. As you can see that DNA molecules, DNA fibers are highly condensed and also highly bent. So the bending and condensation of DNA is really important, fundamentally important in biology or in epigenetics. And the fundamental unit of epigenetics is nucleosome, which is a complex of histone complex protein wrapped by about 200 base pair DNA. So each chromosome carries about millions of nucleosome. It is a kind of pigeon string model of chromosome. And the DNA sequence, there are 2 variables, DNA sequence, and the chemical modification of DNA and histone tail. Those define the function of nucleosome or function of DNA or function of gene in general. How epigenetic modifications and sequence define function of genes. For example, let's consider DNA fiber conformation, which is in highly condensed conformation that looks like that. In this conformation, DNA fiber that is shown as a blue line here is not accessible by RNA polymerase that reach DNA information to generate messenger RNA. So because RNA polymerase cannot bind to DNA here, genes encoded in this domain is inactive. So to activate inactivated genes, first chromatin fiber must be decondensed. By decondensation here, as shown here, DNA became more accessible by RNA polymerase, but it is not fully activated because still there are many histone core proteins are bound to DNA, and those histone core proteins protect DNA sequence information against RNA polymerase. So for each to fully activate gene information, you need to unwrap DNA from histone core complex. As shown here, DNA is fully accessible by RNA polymerase. In that case, RNA polymerase can bind to DNA molecule to read DNA information and transcribe messenger RNA. So as you can see, in fundamental epigenetics in general can be, from the perspective of physicists, epigenetic question of the epigenetics can be reduced into two fundamental problems. One is condensation of DNA. The other one is flexibility of DNA. How those condensation and flexibility can be controlled by DNA sequence information or epigenetic modification informations. That is a fundamental question. So, so first, DNA condensation. So we wanted to study how DNA sequence information and DNA modification of control condensation of DNA. So we built it is not very small system, but a relatively small system with only one pair of DNA helices. And I didn't show every water molecules, but it is a fully atomistic simulation, including explicit water and ions. And in addition to DNA and water, we, as shown, those small molecules are polyamines that carry +4 charge per molecule. And the model of polyamine is shown here. DNA molecule is highly negatively charged. So when you add cation, polycation like polyamine, polycation can induce DNA condensation. And there are two more reasons that I added polyamine in this simulation. One is because most important reason is that polyamine is known to be present inside our cell at millimolar concentration. And also polyamine is, as you can see, by comparing chemical structure between lysine residue, which is important in histone complex, and the polyamine structure. They are chemically analogous each other. Both of them have a functional group, amine functional groups. So polyamine it can serve as a model molecule of lysine peptide. So from MD simulations, we can compute mean force between two DNA molecules. And also we can compute interaction free energy between two DNA molecules. So first, we wanted to know how DNA sequence control the binding of interaction free energy between DNA molecules. So in here, I ran two, in our jargon, umbrella sampling simulation to compute free energy, interaction free energy. For this system, I had one pair of DNA with a GC repeat sequence. And in the other sequence simulation, I had the similar system, but with 80 repeat DNA. So for example, this one shows the interaction free energy curve for GC-rich 100% GC-rich DNA. As you can see, the energy there, certainly DNA molecule attract each other, and binding affinity is about 0.7 kilocalorie per mole per ton. It is close to KT, but you have to consider that it is binding energy per ton, 10 base pairs. So the human genome DNA is really long. So when DNA, the binding affinity is proportional to the length of DNA. So that energy is large enough to induce DNA condensation inside our nucleus. But what is interesting was that when we changed the DNA sequence from GC to AT, the binding affinity became twice stronger, which means AT-rich DNA attract each other twice more strongly than GC-rich DNA. So what is the biological relevance? So our human genome project was finished up 15 years ago. And one of the really surprising findings of this human genome project was that our human genome includes so much junk DNA. For example, this one shows the red bar graphs, shows the position of the junk DNA. And what is also known is that junk DNA has really huge AT content. Some junk DNA can have 100% AT content. And all the gene coding regions are GC-rich DNA. So it was really disturbing to us because only humans, eukaryote, higher organisms like a human, contain junk DNA or AT-rich DNA segment. For example, if you see bacterial DNA, any bacterial genome has really high, constantly high GC content. And every DNA bacteria do some genetic function, which means bacterial genome doesn't have any junk DNA. So we must be more advanced organism than bacteria, but surprisingly, our genome includes more junk DNA. So that was the biggest mystery after the Human Genome Project, but it is not mystery anymore because now we know that junk DNA is not actually junk DNA, but junk DNA simply doesn't have a function as a genes. But junk DNA simply have a different function as an architectural DNA. So what people know from the imaging technique is that AT-rich DNA inside our nucleus form very stable cluster, and they and those cluster formation between AT-rich DNA push gene coding G3 DNA into the center of the nucleus. So most of the transcription activity occurs at the center of nucleus, and I think that kind of localization of the gene coding region enhance gene transcription efficiency. So anyway, it is well known the AT-rich DNA domain is really important for structure of chromosome. But what is the driving force of the AT-rich DNA condensation? Has been unknown, and probably what we found can be the fundamental mechanism that drive AT-rich DNA condensation. So next, we wanted to know how DNA methylation affect DNA condensation. So we took G3 CG repeat DNA, and only in one set of simulation, we introduced methylation of DNA, cytosines. And again, when we compute interaction free energy for methylated DNA, we saw that the binding affinity increased by a factor of 2 again when we have a DNA methylation, which means methylated DNA tends to condense more easily than unmethylated DNA. So what is the biological relevance? And for example, in human female cell has 2 copies of X chromosome, but by some unknown reason, only 1 copy of X chromosome is active. This problem is known as X chromosome inactivation. From this image, one can see that inactivated X chromosome is much more condensed than activated X chromosome. And X chromosome condensation is triggered by hypomethylation of DNA. So because of correlation, we expect that probably the physical mechanism that we found can be the fundamental reason why how X chromosome inactivation occurs. So by analyzing and simulation, we can figure out the microscopic mechanism that how the methylation or AT content control binding affinity between DNA molecules. So for G3 DNA, one of the important character of the DNA is that there are major groups. Major groups are really strong binding site for cations. So for G3 DNA, polycation can bind to the major group very stably. But DNA is negatively charged and polycations are positively charged. So DNA condensation can be strong only when polycations are shared by DNA. But G3 DNA doesn't share polyamine. So that's why G3 DNA doesn't attract DNA each other. But for AT-rich DNA, thymine has methyl groups. Methyl groups protect major group from the polycation. So for AT-rich DNA, every polycation stay outside the DNA. So those free polyamines can be shared by multiple DNA molecules, inducing attraction. So what about the methylation of cytosine? Methylated cytosine is very similar to thymine chemically. So methylation of cytosine also repel polyamine, polycation to outside of DNA. And again, by the same mechanism, the free shared polyamine introduced enhance attraction between DNA molecules. So our prediction from ND simulation was confirmed by single molecule experimental collaborators, Professor Taekji Pa group. So they, it is called FRET technique. I'm not going to introduce FRET technique, but that is very popular technique that can measure binding affinity between two separate molecules, single molecules. So they measure according to their measurement. When we have G3H DNA unmethylated, binding affinity was really marginal. But when they changed that DNA sequence to A2H DNA, binding affinity was significantly enhanced, consistent with our MD simulation prediction. So, and also they also took G3H DNA and introduced methylation, and they also confirmed that enhance of DNA condensation by DNA methylation occurs experimentally. So I want to change the topic to DNA flexibility. For Taekji Pa group also performed the DNA flexibility measurement. From this experimental measurement, they can, by using single molecule FRET technique, they can count how many DNA molecules in the slide are in the loop conformation. When the count of loop conformation is proportional to number of flexibility of DNA, the flexible DNA tend to form DNA loop. So by counting the number of DNA loop, they can quantify which DNA is flexible and which DNA is not flexible. So they, for example, in this sequence, they took a DNA sequence, but with 4 different 4 CPG sites, and they measured using single DNA looping loopability, flexibility, and for unmodified cytosine, the black line folding DNA looping occur like black line here. But when they introduce methylation methylation to CPG sites, they observed that DNA flexibility was decreased significantly. And DNA methylation is one of most popular, well-known DNA modifications, but there are some other types of DNA modifications. One is DNA formylation. For example, what they found was that when CPG sites are formulated, formylation enhances DNA flexibility. So effect of formylation is opposite to effect of methylation. Also, they found that hydroxy methylation has a similar effect to formyl group, but the magnitude is slightly smaller. And also, they found that carboxylation of CPG site doesn't have not so huge effect to DNA flexibility. So to figure out what is the microscopic mechanism that is of the DNA flexibility modulation by cytosine modifications, we took the exactly same DNA sequence and ran MD simulations of DNA sequence. And from that simulation, we found that from MD simulation, we can tell which DNA stacks are most stable by measuring structure standard deviation. Standard deviation is inversely proportional to force constant. So for flexible DNA step, we see larger standard deviation. And when we measured, our simulation result was quite consistent with the experimental measurement. And by analyzing MD simulation data, we could figure out there are three competing components that determine DNA flexibility. For example, first, hydrophobicity decrease flexibility because it blocks the motion of nucleotides, and increase in polarity has the opposite effect to increase in hydrophobicity. And introduction of the additional charge also decrease interaction flexibility, because DNA flexibility mainly comes from electrostatic repulsion between negative charges. So if we introduce a negative charge, then DNA becomes more stiff. So DNA methylation just increase hydrophobicity. So that's why the DNA methylation decreased overall flexibility. But unlike methylation, formylation doesn't increase hydrophobicity, but it just increased polarity. That's why formyl group increased flexibility. Hydroxymethylation also increased polarity as large as formyl group, but hydroxymethyl is slightly hydrophobic. So that's why we see overall smaller increase in DNA flexibility. And carboxylate group is really polar, but carboxylate group introduced additional negative charge. So that increase in negative charge compensate effect. So that's why carboxylation doesn't have any effect. And the final thing I want to show is that we have improved the non-bonded interaction parameters. What we found for the past 5 years is that in both Amber and Chern-Force field, non-bonded interaction, especially for interaction between charged groups, positively charged and negatively charged groups, their attraction is significantly overestimated. And also we figured out that hydrocarbon-hydrocarbon interaction, which is important for hydrocarbon collapse, for example, in protein simulations, that attraction is highly overestimated. So the typical example showing the importance of the improved parameter is from the DNA simulations. For example, experimentally it is well known DNA-DNA interaction should be like that blue curve here. But if you that is actually published data in 2008. If you compute interaction free energy between two DNA molecules using standard Amber or standard Chern-Force field, you get this curve. So it is not just quantitative disagreement, but it is qualitative disagreement. When in the situation that we have to see repulsion, we see attraction. So this kind of, but by applying our improved non-bonded parameters, we can reproduce experimental data. So this is improved parameter set that we named CU-fix. It's really important for the successful prediction in our MD simulations that I showed in the previous slides. And we also performed many, using Blue Waters, really extensive validation simulations for protein folding and DNA protein interactions and lipid protein interactions. And we have a really convincing data showing that correction of the non-bonded interactions are really essential for all classes of protein biomolecular simulations. So everything shown here was essential. Blue Waters was essential for everything I shown before. But what is really exciting about Blue Waters is that currently we are running really massive MD simulation of DNA loop, not a few DNA loops, but we are simulating thousands of different loops and taking with the DNA sequence taken from DNA genome. So in collaboration with the Ha group, what we are trying to do is making a genome-wide profile, DNA flexibility profile, and that will be really important for epigenetics. So I'd like to thank my funding agency NSF and the Blue Waters, and my collaborators, and my advisor and my group member, Chris. Thank you for listening.